He says I'm good to go. All right, so let me bring my first slide up here. So that is the title, but that's sort of a joke, and I'll tell you a little bit more about why it's a joke in just a minute. So the real important title is congratulations, Jerry, on a very well-deserved award. And I'm really pleased to be here because Jerry asked me. So about 5 years ago, Jerry got another award at Pittsburgh. I forget exactly which one it was, but it wasn't snow, it was thunderstorms. And I missed my plane. They canceled the plane, and I didn't get here. And I got in trouble with the Pittsburgh people. They were mad at me for not being here. I was banned for like 5 years or something. Anyway, so I figured I better come this time. But I'm sure a lot of you are like, Sharon Neal, what is she doing on a symposium with Jerry? Isn't she a vibrational spectroscopist? Sharon Neal doesn't know anything about vibrational spectroscopy, and that's almost true. But I'm going to talk to you about this project. So when Jerry asked me to do this, I started thinking about, well, what am I going to talk about? So I'll tell you more about how I arrived at this project in just a minute. But of course, for these awards, you know what you're supposed to do. You're supposed to show all the pictures you have of the awardee. So I went onto my hard drive, and because I'm on the advisory board of Coach, which is a wonderful organization, and I'm so pleased to be a part of it, I have a lot of photos with Jerry and I. And is there a laser pointer? Yeah, I think here it is. The one I really want to point out to you is the one where Jerry's hamming it up over here. just like Bruce pointed out, she does have a humorous side. So for all this accomplishment, and Jerry is very accomplished, and amazingly so, in my opinion. She's a wonderful human person and a warm person, and I'm really appreciative of being invited to participate today. And one of the other things I think people do in these settings is they say a little bit about their interaction with the person. And so most of my work with Jerry has been with Coach, but even maybe a little bit after we did Coach, I was present at a workshop that Jerry was one of the main organizers for. This was the Gender Equity Conference that was held, I believe it was back in 2006. And one of the things I saw there that impressed me quite a bit, this is a panel that Jerry ran with, this is President Emeritus Vest of MIT and some late Chancellor UCSD, Denton, some very prominent people. But the important thing about this symposium is that it was really well motivated by the literature. And one of the things that I think is really important in this area of expanding participation in science of underrepresented groups is that we have to, a lot of times there are a lot of myths around why it takes so long and why it's so slow and why it's as hard as it is. And I think one of the reasons we don't make the progress we could be making is that we don't apply our scientific method. We don't use the same strategies that we use to solve tough scientific problems to address this problem. We have an idea off the top of our head. We have myths about people with not wanting to do these kinds of jobs and preferring to play basketball. Just to tell you something that someone actually said that to me once, unfortunately. And instead of studying it, we have these ideas in our head and we kind of act out of those. But Jerry showed me that when you think through your strategies, you base them on people really studying the issue and being scientific about it, you can come up with some strategies that actually really address the issue in a meaningful way. These are tough problems. They're not going to go away quickly. They didn't evolve quickly. They're not going to be solved quickly. But you can make real progress. And so here's a little chart from CNE News just showing how the number of women on chemistry faculties is changing. And what I really like about this is the headline. It says, "Isolation Diminishes," because that is really one of the main detrimental impacts of the lack of distribution of women and underrepresented minorities in chemistry, is that individuals who could and should flourish and would enrich the lives of everyone around them find themselves isolated because they're in these situations where people haven't really thought about, well, how do we incorporate this person into our body politic? So that's some of what I learned from her. I'm still trying to learn to put all that to good use, but it's really a pleasure to be here. And I wanted to say to you and say to her that she's doing wonderful work, and I hope that she'll just continue for many, many years. I don't know what other awards there are left to give her. I guess the Priestley Medal and the Nobel Prize, right? That's about all we have left. Okay. So now to the story about where this title comes from. So as I started thinking about what am I going to say at this symposium, one of Jerry's most cited, I went to Web of Science, of course, where we all go. One of Jerry's most cited papers is Water at Hydrophobic Surfaces, Weak Hydrogen Bonding, and Strong Orientation Effects. And so I thought to myself, okay, how do you take some project that you've done and kind of reorient it and make it fit into that framework? And so that's where that title of Lipids in Water Aggregates Temperature Effects comes from. It's a joke. It's a play on these words, because I'm not a vibrational spectroscopist, and I don't pretend to be able to actually do these kind of things. So I wanted to start out with that disclaimer, and I'll also tell everyone in the room what those of you at the back know is that my computer has been crashing every few minutes. So hopefully we'll get through this without me having to show you that I can be poised under very serious duress. So I have a story to tell you, and I want to motivate it with a little bit of a backstory. So when for many years, so very early on in my career, I was always interested in self-assembled systems and non-covalent association and interactions. And so I've always sort of been interested in vesicles and micelles and so forth. And so very early on, I learned about this really interesting biomembrane mimic called bicelles. And so here's a classical picture of the bicelle, the long chain phospholipids oriented in a bilayer and then end-capped on the ends with these short chain phospholipids. And there was some really interesting work showing of their utility in measuring solid phase NMR spectra of integral membrane proteins and the like. And the model that people had back in 1998 about why they would orient in a magnetic field and therefore allow that solid state NMR spectrum to be collected was something like this, that the individual bicelles would align with the field and trap water, and that's where it was all coming from. One of the things they didn't say was that these systems become very viscous as you heat them. And so, I'm an analytical chemist, I'm a spectroscopist, I'm used to putting very small amounts of molecules in solvents, and I'm not used to this idea of, putting something, not that much, but putting something in water and you heat it and it's solid. And so I had a student, a good student at the time, and we decided, and I'll tell you more about these experiments in just a minute, but we were going to put fluorophores into these mixed lipid systems with the idea that these are membrane probes and their spectroscopy would change, and I'd be able to do these really cute numerical separations of the different components of the spectroscopy. That was the whole idea. And so the student mixed the sample, and he puts it in front of the laser, and air bubbles are generated and photons start flying everywhere, and he runs to my office and he goes, you need to come see this. And so eventually, so I said to him, you must have done something wrong. This solution must be dirty. There's some contaminant in here. You do it again. Of course that's what you say. And so he did it again. And the same thing happened again. So finally I go and I stand right next to him and I watch him. And I'm like, oh, this kid's hands are better than mine, right? Because I'm the computer geek and I drop stuff, I break stuff, I cut myself every time I'm, near glass. That's actually serious. And so I realized that wasn't a problem. So I went to the literature and I realized, I learned later that one of the things they weren't advertising was that these systems do become very viscous. And so I was really intrigued by that. And I was like, well, what are we going to do? How are we going to figure out what that is? And so we arranged this FRET experiment, or this is the plan, where we tag the long chain lipids and we tag the short chain lipids, and the short chain lipids were tagged with my friend Pyrene, and I had done a lot of work with Pyrene before that, so I felt really confident that I'd be able to separate the spectroscopy of the Pyrene monomers from the Pyrene Xmers, and then the Pyrene Xmers were going to have this interaction with the long chain probe, and we were going to be able to watch the evolution of the spectroscopy that we thought would occur as these mixed micelles went on to form bicelles. Actually, it starts here and goes up, sorry. So this is what we thought would happen, and of course we took control spectra and saw the spectra that we thought we were going to see, but when we actually did this experiment and tried to measure lifetimes as a function of wavelength, we never saw. We actually all of the emission from the Pyrene went away. But one of the interesting things that did happen as I looked at this data more closely, even though it's very small, I was able to track. I actually was able to pull out a very small Pyrene spectrum from this data and track its behavior. And what I saw happen was that small Pyrene spectrum, there's no FRET here. This is just as a function of temperature. That small Pyrene spectrum got disrupted and then reappeared. And I wondered, my goodness, what is that? How could that happen? What is that about? And I thought about it, did a little bit of reading. It wasn't established, but it was clear that the only way that the data that we were seeing made sense is if these disc-shaped bicell units were coalescing into something with a perforated sheet. And so we wrote that paper and we said that's what happened. And not too much longer after that, this is obviously a long time ago, there was small angle neutron scattering data that said, in fact, there were perforated sheets being formed. And so I thought, oh, this is really cool. This is really exciting. I really want to study these systems. I've got to find ways of getting more information about what's happening with them. One of the other motivations, and I don't think she's here, I know she's in the building, but Lisa Holland and I were talking by phone, and she did this really interesting work where she put these mixed lipid aggregates into these microfluidic channels, and she was actually able to make thermal actuators and push liquid around on these microfluidic chips by changing the temperature of these lipid mixtures. And so when the lipid mixtures are cold, everybody flows, everybody moves, you heat it up, and now you got a barrier, and this happens a whole lot more quickly than what you could do with a pneumatic actuator, right? So that's the exciting part about this. The downside is that it was hard to control, and she may be doing better with this now than she was at the time, but one of the problems that she and I talked about this, and you make these solutions and you make them the same every time, but in my hands, in our lab, maybe about 10% of the time, the solution would just fall apart for no good reason. It would just phase separate. You don't know what's going on. For her, in these microfluidic chips, she said that this phase separation and the solution falling down would happen in about 30% of the time. And so we hypothesized that probably shear forces at defects on the surface of these microfluidic chips were probably were initiating these phase separation events. In any case, we never did really get to study that phenomenon in detail, but that's what got me looking for other ways to understand what was going on with these lipids. And so I'm telling that the main story today is this cute little story, and Professor Harris is in the back, and so I was reading one of his papers. So he can do these great Raman experiments on drops, right, and beautiful spectra from drops. Go figure. Anyway, or individual vesicles levitated in front of the laser beam, but his data shows, and lots of data from way back, shows that you can monitor the behavior of the alkyl chains in lipids using Raman spectroscopy very well. And so I was thinking, well, you know, I've got access to a Raman spectrometer. I'm just going to put this system in here, and I'm going to monitor it as a function of temperature, and I'm going to see what happens. And so these are Raman spectra that were collected from 3-to-1 mixtures of long chain and short chain. The long chain were DMPC, that's 14 carbons, the short chain 7 carbons, collected as a function of temperature between 7 and 42 degrees, and then arranged in matrix format. So here's the raw scattering melting matrix, and you can see the CH and stretch and bending bands sitting on top of this very round spectrum. And I'll tell you another joke. Make sure you, well, I can't make sure, but if you're sleeping, maybe this will help. Anyway, so a few years ago, Professor Mendelson at Rutgers, who's really well known for Raman spectroscopy of lipids, came and visited our department, and he saw this data, and he said to me, "Oh, your lipids are very clean." And I thought he was serious. And so many years later, I looked at this and it hit me that he was pulling my leg. He was saying, "You got lots of contaminants in here, girl." And I didn't realize what was happening. But I didn't realize that he would joke with me. And so that's what happened. Anyway, he was pulling my leg. So we've got Raman bands sitting on top of something. So I've been doing this a lot of years, and whenever I try to talk to people about the mechanics of the numerical separation that we do here, I kind of get glassy eyes and even more sleeping than before. So I'm going to spare you that. I'm just going to tell you a little bit, okay? So the main reason why you want to arrange these spectra into a matrix and go through the pain and bother of learning about matrix algebra, if you don't like it, like some of us are weird and we like this stuff, but for everybody else, the real reason why you want to do this is, first of all, you get an opportunity to get an estimate of the number of components in your sample without having to do anything other than just count the number of components. And so here what I'm showing you is the data matrix. Now remember, these matrices are products. Just like 12 can be broken up into 3 and 4 or 2 and 6. If you have a matrix here, we've got spectroscopy on one side and temperature behavior on the other. That means that we're going to be able to break this table of data up into some data that says something about the spectroscopy and another set of data that says something about the temperature behavior. And so that's all we have going on here. And what's important is that these factors are mutually orthogonal, and so we can, it minimizes the number of these components that you need to reconstruct this data. And so when you have noise superimposed on the spectra, as we almost always do, you end up having as many components as you have the smaller dimension of your data. But only the first few of them actually have any real spectroscopic information in them. And that's what we are attempting to count here. So I've got a couple of statistics that are telling me something about the amount of variance associated with each of these components, and more importantly, what's the word I'm looking for here? I'm missing a word. The frequency content, the relative importance of high frequency contributions to these components. And so these early components, they're mostly spectra, so they're mostly low frequency. We all know that. That's why we use filters, right? And so these first, say, 5 to 7 components are the ones that have spectroscopy in them. So as I said, I'm going to not bore you with the details of how you estimate spectra from the matrix. If anybody's interested in that, I'll talk to you about it after. What I will tell you is that this is a little bit different than some of the other curve resolution, self-modeling curve resolution methods you might have heard about. What I tried to do here was a generalization of a method used in electrical engineering called the Ho-Kalman method. And so the reason why that's an important thing is that I had tried to do this using traditional methods, including one that I had written myself as a graduate student, and this contamination in this background was making it really difficult for me to isolate the spectra and separate the spectra from the background. And so this new method that I will not talk to you about seems to be doing a much better job of that. So here's the overview picture. There are spectra, there are temperature profiles. Let's look at them more closely. How am I doing time-wise? I tend to talk really fast when I'm nervous. And I'm more nervous today than usual. Some of that is because Jerry's so important and everything. But some of that is, you know, often when you're doing these, that you're talking about stuff that you've given this talk like 5 times. You know, you've been out on the circuit, you've given it to like 5 or 6 different departments, and I've never talked about this before, so I'm going kind of fast. I'm going to take a deep breath here. Okay. All right, so let me talk to you about this data a little bit. So as I told you, it finally hit me that, you know what, this is not very clean lipid samples. I got them from Avanti, who's usually very wonderful, but for whatever reason, there's a component very broad. This is fluorescence, probably NADH or something that's in with the eggs. And what's interesting to me is notice that we've got really wonderful sigmoidal behavior, which I was really surprised at. But when you think about it, that makes sense, right? You heat the sample up and the fluorophores get quenched. And so maybe that's not so much of a surprise. One of the things, the other sort of background component is sodium azide, which is added to inhibit microbe growth. It probably wasn't really needed because we made this sample in a NMR tube and then we had the glass blower seal it shut. So no microbes were getting in there, but that's sort of a standard part of the preparation for these bicels. Avanti sells the kits and has the procedure, and you get in the habit of just doing the procedure even when you don't need it. So it's here. One of the reasons I wanted to stop and talk about this, though, you see how the temperature response here is actually going below 0 in a place. So what that tells me is that even though I'm doing really better than I was on isolating the background and getting it out, I'm still working on this question of how many components are there. And the work I'm showing you today, this is the analysis with 5 components. Once you go to 6 and 7, you're introducing more noise into your spectra, and it's more complicated because you got more components that you're fooling with. So anyway, that's why we're on 5, but this is one of the pieces of evidence that I still have a little further to go. I still need to work on getting all of the spectral responses and all the temperature profiles exactly right. So I think these are close, but there are some places where it can get better. So what's going on with the lipids? And remember here I've got short chain lipids and long chain lipids, and that's all in this solution. So I've got the 7 carbon and the 14 carbon, and we watch them as a function of temperature. The temperature is increasing. I was able to separate the lipid response into 3 spectra, And of course, with time, I'll be able to just rattle off what they are. But obviously, this is CH stretch. We all learned about that in organic. But you can see that lipid number 1 is decreasing, number 2 is increasing. So you think, okay, well, this one's decreasing. Maybe that's the gel phase. This one's increasing. That's liquid crystalline. But then there's another component that's decreasing, and what is that? So I'm still learning about spectral assignments, but I've got some model spectra here for the CH bending and CH stretching. And in the gel phase, you see, first of all, my eyes are no longer good enough to read that far, and I'm sorry, but you can see that the relative height of these peaks here in the bends is different. These two are much taller here in the liquid crystalline than the gel phase. And then when we go over, actually I think those are the stretches and these are the bends. Here we've got this really clear triplet, but then in the liquid crystalline phase, these two bands have dropped, and they're around the same relative intensity as the center band. So those are features that we can use to tell what are we looking at. And so for our blue spectra, well, for our purple spectra, we can see that's definitely gel phase, right? So that third one is the one with the classic gel phase structure. The blue, the green, right, looks pretty good for the liquid crystalline. And the blue is an interesting, it's got the liquid crystalline CH bend, but then when we look here at the stretch region, we've got that tall center band that we associate with liquid crystalline, right, on this one that has gel behavior here. So we've got this really interesting kind of mixing up of the response. The other part of the spectrum is the part that Jerry and other people here know a lot more about, and that's the water bands. And so what we have here is that in that first blue spectrum, we've got a really blue shifted water spectrum. We've got the traditional liquid crystalline, and here's our reference. And so at 50 degrees, this is what you expect your liquid crystalline water to look like. The dotted spectrum is buffer, and the black is the lipid. So we've got our traditional liquid crystal for the green. Our blue is very blue shifted, which suggests to me that the water here is very ordered. And then the purple, the magenta, is very similar to what we see at 10 degrees, looking very much like the traditional gel phase lipid spectrum. And so what we see here is a set of lipids that behaves has spectra very consistent with what's been seen before for the gel phase and for the liquid crystalline phase. And then this interesting third phase that has kind of a mix, a very ordered water spectrum, but then when we go back and look at our CH spectrum here, this we would associate with liquid crystalline. So it's kind of interesting. So while it doesn't provide the interface selectivity, multivariate analysis does allow you to separate these into responses of the components of complex mixtures. It was able to isolate and enable characterization of the lipids, but just as importantly, we were able to isolate and pull out the responses of the background. And that's an important thing, because earlier in Bruce's talk, I think I heard you say there's something, we've got a contaminant going on here, we'd like to be able to pull it apart. And so this is one of the valuable features of this type of analysis, that many of the components that we aren't interested in are still well characterized, and we can kind of put them to the side and go on and look at the other components. Unfortunately, the real big thing that I was looking for was some signature related to those morphological changes, the water gelation, and the interesting high viscosity. And I didn't see any evidence of that in the Raman spectroscopy. So I think the moral of that story is that you have to go to terahertz spectroscopy to see stuff like that, because these aren't molecular kinds of interactions. They're big lattice level things, and I think that's the moral of that part of the story. So with that, I'll just acknowledge the students who actually did this. Both of them have both graduated and gone on, Brad and Eric. Eric actually acquired all of this data while he was doing a rotation with me. He was I forget the name of our NIH-funded training grant program, but that's what it was. And so that's the building where a lot of the data was collected. And way back when a lot of the data was collected, the National Science Foundation, and I'm supposed to know better than not to. I'm supposed to know that you have to put the award number on here. And so today I'm here as a University of Delaware faculty member, but I'm just finishing 2 years as a rotating program officer, and I still do some part-time work, and I missed that as I was going through these, so I apologize to you. I'm not supposed to do that anyway. So just so I made it to the end a little faster than I was supposed to. So I imagine that there were details I intended to tell you that I went too quickly to do, but of course I'll be happy to answer any of your questions. And we're going to break after this, so that means that I'm actually giving you a few extra minutes with your coffee, so you can thank me later. Thank you for your attention.
Questions?
Christy.
I'm interested. I also like data processing, so count me as well. It sounds like you're using an information theory method to calculate maximum likelihoods of the numbers of spectra that you have. Is that what you're doing?
Not quite. So, I'm sorry, not quite. So, actually what I'm doing here is I built the three-way Hessian, the three-way Hankel matrix for this, because the temperature transition, and actually I started sort of at the middle of the sigmoid. And the idea here is that every step is approximates a linear step. And so we're going to generalize the measurement by describing it as a system of linear equations, not just the decay, but the whole acquisition, including the excitation and so forth. And I was able to set up this more exhaustive set of equations that I could solve for the spectra and the time and the temperature profiles.
Can I ask, what was the hydration level of those measurements?
So these are 25% lipid, 75% water. The Q ratio is 3:1, 3 long chain lipids for every 1 short chain, and that was selected because that was the recipe a lot of the protein spectroscopists were using to prepare these systems that they were using for NMR of the membrane proteins.
And were these dominantly lecithins? Dominantly phosphatidylcholine?
Yes, they were all phosphatidylcholines. 14 carbons and 7 carbons, all PCs.
So if I remember right, maybe I'm wrong here, I remember that in the alpha crystalline phase, that the maximum hydration was like 40%. Is that?
I'm not, I don't think that's, and I don't think so.
I could be wrong. It's a long time since I
Yeah, for
read
me,
that.
yeah.
But the question is, did you have a two-phase system? Because
Mm-hmm.
when you had some spectra, I think, at a higher temperature, where the water Raman spectrum was very different,
Right.
right? It didn't look like any bulk water.
Bulk water at all, exactly. And so was it a, so it certainly wasn't, it didn't phase separate, for these samples, that didn't happen. Whether or not there was some different kind of organization of a subset, that's what we have to understand. And once these spectra are completely assigned, I don't. That's the question. That's the question, right? I don't know.
Other questions? Well, it's time for our break. We should reconvene here at 3:40.